Okay, I think we're going to start. There are many good reasons to go to Houston. One of them is barbecue, and the other one I can think of is Jim Lupski. And let me tell you a little bit about him. I have a huge list here. He got his MD, PhD from New York University. He did a residency in pediatrics and a fellowship in medical genetics at Baylor College of Medicine, where he is currently the vice chair of molecular and human genetics and also the head of the medical genetics laboratory. And he has numerous awards. I picked out a couple. The Kurt Stern Award for the American Society of Human Genetics, and he was elected to the National Academy in 2002. And apparently he has 430 publications that bear his name, which makes me think that there's more than one of you. So today we have a fascinating subject. You're going to hear a lot more about the types of genomic rearrangements that are hidden from the DNA sequences and they're hidden from the cytogeneticists, but they've been revealed to be very, very common through microarray hybridizations. And you're going to hear a lot about this as a general theme for genomes and also clinical implications. So thanks for coming, Jim.
Thanks a lot, Neil. Yeah, if we could get those lights down, that would be terrific. Well, I want to thank Neil and the faculty and the students for inviting me to come here and for the opportunity to share some of our work with our colleagues here in Davis. So as Neil alluded to, I'll be talking about structural variation of the genome. Actually, I think I'm going to need these lights out. The back ones you can, because I'm going to show fish pictures. It's hard to see. I think it's those spotlights. We have to get out somehow. Oh, now we're talking. Beautiful, beautiful. Terrific, terrific. Okay, so if we think about human genetic variation, actually this is a picture from the cover of Science from last year when human genetic variation was chosen as the molecule of the year. And interestingly, up until just a couple of years ago, we didn't appreciate the fact that we vary a lot in copy number of different segments of our genome. And if everybody in the audience just looks at the person next to them, you actually each vary from each other more in terms of number of base pairs that have copy number changes than all the single nucleotide polymorphisms in the genome. A significant percentage of the human genome has copy number variation. And in fact, some 15% of genes listed in OMIM, online Mendelian inheritance in man, causative for human disease, are involved in different copy number regions of the genome. So today I'm going to talk about this structural variation and talk about it from the perspective that our lab had come to this through the study of so-called genomic disorders. And we'll talk a little bit about the rearrangement mechanisms, recombination mechanisms, a non-allelic homologous recombination, briefly touch on non-homologous end joining. I don't really have anything new to add there. And then spend the majority of time on something called, we've termed fork stalling template switching, as a DNA replication mechanism for genomic rearrangements. Then in that context, I'll talk about how we've designed clinical arrays to bring these into the clinic and look for genomic rearrangements as cause of disease, and in particular with its relevance to birth defects, which are mostly sporadic traits in human beings, and look at the frequency of de novo mutations that are associated with sporadic birth defects. Then I want to also just briefly talk about a forward genomics and reverse genomics approach to delineating the clinical consequences of copy number changes, and something we refer to as, rather than genotype-phenotype correlations, genomotype-phenotype correlations. And then briefly, again, about de novo rearrangements and birth defects in neonates, so you get a flavor for its relevance to the clinical situation. For the replication mechanism, we recently published Jenny Lee, who's a graduate student, and she did a terrific job after 2 extra years of beating her head to finally figure out what some of these breakpoints might be telling us. She was assisted by Claudia Carvalho, a postdoctoral fellow, but I'm going to talk mostly about Claudia's work on MECP2 duplications, none of which is published yet. And then Feng Zhang from the Shanghai Genome Center at Fudan University has done a beautiful job as a postdoc, and I'll talk about this REI1 dosage changes and how we use this to assay different rearrangement formation. And for the other parts of the talk, relating to the genomotype information. Zhang Yang was a postdoc in the lab, Weimin Bi, a postdoc, and Jinyan Liu, a postdoctoral clinical fellow in clinical cytogenetics, and how this is being how she helped translate some of this into the clinical laboratories. So genomic disorders. About a decade ago, a little over a decade ago, I had put forth the concept that many human traits, many diseases in particular, which this is what brought patients to the doctor, were due not to sequence-based changes, but actually occurred because of genomic rearrangements. And we put forward the idea that, in fact, the architecture of the human genome was such that it may have susceptibility regions. Certain portions of the genome may be more susceptible to genomic rearrangements, and this could be responsible for recurrent events occurring over and over again. So that was sort of the concept of genomic disorders. Now, when we started to look at genomic rearrangements, we saw different patterns. One was a recurrent event where we always saw the same breakpoints, and this is so-called breakpoint clustering. And the breakpoints clustered in low copy repeat sequence elements. The next type of rearrangement we saw we called non-recurrent, because the breakpoints are all different in different patients, and the sizes of the rearrangements can all be different. But we do see one region of the genome, and this is the smallest region of overlap, which is shared by patients with the same phenotype, and it seems to encode the dosage-sensitive gene or genes that are responsible for conveying the phenotype. Only a subset of genes in the human genome seem to be dosage-sensitive. Then the third kind of data that we started to see was something we refer to as breakpoint grouping. It was different from clustering. We didn't see all the sequence end in a common low-copy repeat sequence, but instead we saw that they were non-recurrent events, but at one end we saw grouping of the breakpoints. And these were the three sort of types of rearrangements that we started to observe in multiple different diseases, and then we went and tried to sequence breakpoints to infer mechanism from these studies. Now, we focused initially in the proximal short arm of chromosome 17. You got to remember, some of these studies were done 15 years ago. We didn't have a genome sequence. Every single patient was a genome project to try to figure out where breakpoints were. And there's four conditions that map in this region. One is Charcot-Marie Tooth disease, a common inherited peripheral neuropathy, and it's due to duplication of a 1.4 megabase segment. The reciprocal deletion causes a disorder known as hereditary neuropathy with liability to pressure palsies, or HNPP for short. Suffice it to say that we know there's 26 genes in this segment, but this is the only dosage-sensitive gene. We can find patients with HNPP that have point mutations in PMP22, and they're always loss of function or frame shift. The loss of function alleles are the nonsense or frame shift mutations. While we can also find point mutations in non-duplication Charcot-Marie Tooth disease, and they're gain of function point mutations, like we see the mechanism for duplication. More proximally, there's a cytogenetically visible disorder known as the Smith-Magenis syndrome, named after Ann Smith and Ellen McGinnis, and it has common breakpoints. We showed the majority of patients have common breakpoints, and it's a recurrent rearrangement. Occasionally, we find patients who have a larger deletion, and it encompasses, it utilizes a different set of low-copy repeats for recombination substrates, and I'll talk to that in a little more detail in a second. We had predicted a reciprocal rearrangement to this deletion, and then many years later described the duplication, and now actually it's called the Bataki-Lupski syndrome associated with that specific duplication. The frequencies of the recurrent events when we look at families are shown here in the right. So as we map more and more patients, and you look at, say, 100 families with the Smith-Magenis syndrome, we find this recurrent rearrangement about 70% of the time, this common but an uncommon recurrent rearrangement, and then this series of uncommon and non-recurrent rearrangements. And I'm going to focus, I'll talk a little bit about these, and then I'm going to focus most of the talk on the non-recurrent rearrangements. And likewise for the duplication, we find this recurrent, common recurrent rearrangement. Now, we would predict we should find the recurrent uncommon duplication. We've not found it yet, but I think it's just a matter of numbers. We're only up to about 40 families at this point. And why do I say that? Because by single sperm PCR assays, you can actually show that that occurs at a certain frequency. We just haven't found the clinical phenotype yet. I think it'll be just a matter of time. And then we get these non-recurrent rearrangements. Now, how do we know? Now, I don't know why I'm missing some of the figure on the left side. If a person, maybe an AV, can figure that out, that would be helpful back there. But how do we know that the REI1 gene is the dosage-sensitive gene in the deletion and duplication syndrome? In the deletion syndrome, we can find rare patients who have the Smith-Magenis syndrome and they don't have deletion. And then when you sequence, and Sarah Elsia nicely did this in a paper in 2003, you find that they have point mutations in the REI1 gene. This is a zinc finger, actually it's a ring finger gene. And it interestingly has a polyglutamine repeat with a trinucleotide repeat. And this polyglutamine repeat polymorphism has been associated and linked to response to neuroleptics in schizophrenic patients and age of onset of spinocerebellar ataxia type 2. Most all patients with Smith-Magenis who don't have deletion instead have point mutation. This is only about 2 to 5% of patients. They have frame shift or nonsense alleles. We see an occasional missense amino acid substitution whose functional relevance is still a little bit challenging. How do we know this seems to be the dosage-sensitive gene in the duplication syndrome? We don't have any human patients yet with point mutations that seem to be gain-of-function point mutations, but instead we engineered mice using CRELOX-P and chromosome engineering system that was developed by Alan Bradley while he was at Baylor. And we made animals that were duplicated for the 3 megabase segment that's syntenic to the SMS region. Then we mated the duplication animals to the deficiency animals, and these animals have completely normal phenotype, while the duplication animals have a phenotype with many overlapping features to the Potocki-Lupski syndrome. The deficiency animals have overlapping phenotype to the Smith-Magenis. Then we mated the duplication animals to the REI1 knockout allele, and these are very interesting animals because they're trisomic for 40 different genes, but they reduce the copy number of REI1 to 2, and we can completely correct the mouse phenotype, including the obesity we see in the deletions or underweightness that we see with the duplication, the behaviors that we can objectively characterize using mouse different experiments, and the learning and memory deficits. This all gets corrected when you just correct the dosage of that single gene, even though there's some 40 genes in the interval. So now these are the assay systems. The patients then have the phenotype. We can find the event at that region of the genome, and then we can start to study the events. And the 3 mechanisms that have been found, or evidence for them through analysis of breakpoints, has been non-allelic homologous recombination, non-homologous end joining, and the fork stalling template switching, which is the most recent model that has been put forth. So let me talk about non-allelic homologous recombination. We started to see many of these different diseases with recurrent breakpoints mapping to low copy repeats, and 5 to 10% of the human genome has these low copy repeats. They're not repetitive sequences. They don't, in reassociation kinetics, drop out like ALU and line sequences. There usually is only 2 copies or sometimes 3 or 4 copies of these low copy repeats. And they're highly sequence identical, 98 to 99% identical for the majority of parts. Now, if these low copy repeats are in direct orientation from actually, just like it was proven in bacteria many, many years ago by John Roth and others, you get deletion or duplication depending on which way the strand exchange goes. If they're in inverted orientation, we follow the crossover ACBD, you invert the segment that's in between. It's a little bit more complicated in the human system because, again, you've got 2 chromosomes, and it depends on whether you have an interchromosomal event, an interchromatid within one chromosome, or an intrachromatidal event. So you can get deletion by all 3 means, but duplication you can only get by interchromosomal and interchromatid. And we do see in single sperm PCR assays, Matt Hurls has published a beautiful paper in Nature Genetics a few months ago that showed that deletions are favored at an individual NAHR locus over duplication 4 to 1, and the majority of that is due to more intrachromatidal rearrangements versus interchromatid or interchromosome. So why the term non-allelic homologous recombination? I'm accused publicly often of reinventing terms that have been around in genetics for a long time. And they say this is just unequal crossing over. We started in the fruit flies many, many years ago. Why do you need a new term? Well, because unequal crossing over really refers to the products of recombination and how we measure the segregation of marker genotypes. This mechanism speaks to molecularly how the mechanism is occurring. So we require homologous sequences. So if the low copy repeat doesn't have 97 to 98% identity, we never see this happen. It doesn't happen between allels because they're 80% identical. If it's closely related allo families, it sometimes happens, probably because if you start with a normal allo-allo recombination, the mismatch repair system will break it down because there's so many absence of homology regions. So we need homologous sequences. We can demonstrate recombination hotspots. And in fact, we demonstrated recombination hotspots for non-allelic homologous recombination years before they were found for allelic homologous recombination in the human genome. And we can show gene conversion events. This is all clearly part of homologous recombination. Moreover, from the prediction of direct-verted inverted repeats, we can predict, now that we have human genome sequence, which portions of the human genome will undergo deletion, duplication, and potentially inversion. And Evan Eichler's group, who I understand was here a short while ago to give you guys a lecture, he's a terrific person, and he was actually a graduate student at Baylor. I was on his thesis committee. He then used this in computational predictions of the genome to look at the most unstable regions most likely to undergo this. And in the last year and a half, he's described 6 new genomic disorders based on this kind of approach to the human genome. And we've, I'll show you later on, incorporated this bioinformatic analysis into the design of our clinical arrays. And the clinical arrays are picking up more since we did this kind of approach, rather than just an approach of looking at known regions where genomic instability was, and they were defined with a syndrome. Now we can try to predict what are going to be the regions. So the lessons we learned from non-allelic homologous recombination is that genome architecture is important to genome instability. The repeat size is important. Percent identity, distance between repeats, whether they're direct or they're inverted repeats. These are reciprocal rearrangements. Wherever we see microdeletion syndromes, and about 50 microdeletion syndromes were described before a single microduplication syndrome was described, and now for almost all 50, the reciprocal microduplication syndrome has been described. Oftentimes you have a completely different phenotype, or you'll have a different clinical phenotype but the same systems involved. Like Charcot-Marie-Tooth disease, both of these are demyelinating neuropathies, but clinically HNPP presents very different from CMT1A. They're reciprocal, but they're not equivalent. So as Matt Hurls has shown, by examining 6 different regions of the genome at the recombination hotspot for non-allelic homologous recombination, deletions outweigh duplications in terms of events forming during meiosis at about 4 to 1 at every locus looked at. There are recombination hotspots, and interestingly, these can coincide, the non-allelic homologous recombination hotspots with the allelic homologous recombination hotspots, and you can predict genomic regions that undergo duplication deletion. So this actually just shows reciprocal recombination between the CMT1A at the CMT1A duplication HNPP locus. So we can see on this southern blot a similar sized junction fragment in duplication, different size junction fragment in deletion. These are the predicted sizes of the junction fragment. And we see this because even though the repeat's 24 kb, there's a recombination hotspot where 90% of the patients strand transfer. Likewise, we were able to narrow down the crossover for the hotspot in Smith-Magenis syndrome, and then predicted that the duplication hotspot would be in the same vicinity, and we're able to show that. Interestingly, as you can see, it's the same mechanism. But what's very, very interesting is the acceptance in the field of the mechanism. So this, you see, is placed horizontally, because that's the way molecular biologists think. But we couldn't convince the cytogeneticists till we made the model vertically, because they think chromosomes and they think in a very vertical way. So it's very, very interesting. So since this time, we've been going around the genome and applying the clinical arrays, and we can start to find, and for instance here, the Williams syndrome was described for some 25 years due to deletion on chromosome 7, predicted the reciprocal duplication, applied the clinical arrays, and the reciprocal duplication as a case report was reported in 2007, and by late in 2007, we already had 7 different families. Most of them all had the same size duplication. One family had a little bit different duplication. The only presentation was speech delay and autism in these children. This is the diagnosis that they held. And interestingly, 2 of the 7 families, one of the parents had the duplication, but this seems to be a reduced penetrance allele, particularly duplications versus deletions, although both of them had had some speech therapy early in life. Likewise, in early 2008, it's been known that the George syndrome deletion for a long time, and recently the reciprocal duplication had been described about 3 years ago. We described the distal deletion from the George locus that uses alternative low copy repeats, and that was described in January 2008, and then just a few months ago we found all the reciprocal duplications to those predicted deletions with different syndromic features. Again, the duplications were milder or conveyed milder traits than the deletions did. Now, for the recombination aficionados in the audience, during my sabbatical I worked a lot with Matt Hurls at the Sanger Center, and we wanted to address the question of, are non-allelic homologous recombination sequences in the human genome different from allelic homologous recombination sequences? And actually, we could not find any motifs that seemed like cis-acting sequences like chi around any of these. But what Matt's group did is now sequence the CMT1A repeat regions from 100 normal individuals that didn't have Charcot-Marie-Tooth disease. And then from the sequence, you could look at polymorphism, single nucleotide polymorphism. So the proximal repeat had either an A, or the distal repeat in this case had A or G. So this is a single nucleotide polymorphism. Distinguish that from paralogous sequence variants. The proximal repeat all had G, the distal repeat all had A. And then that from multi-sequence variants, where you have variation at both the paralogs and the allelic positions. And actually, you can find your recombination hotspots by an index of the MSVs over the SNPs plus PSVs, and it falls out the allelic homologous recombination on top of the non-allelic homologous recombination. So we believe that in both situations, whether you're doing ectopic recombination or you're doing recombination at allelic positions, there's probably some of the same machinery involved and perhaps some of the same signals that make places recombination hotspots versus cold spots. Now, non-homologous end joining, basically, this audience, I don't need to talk, but basically one has double-strand breaks, and from a breakpoint sequencing standpoint, the key is that you can see a signature. There's no homology on either end, and your signature might be microhomology or addition or loss of bases where the repair takes place at the joining of the double-strand break. Now, when we took first Smith-McGinnis patients that had non-recurrent rearrangements, and again, each one of these is a genome project, so we don't have a whole bunch of information, but we sequenced breakpoint on 4 patients. Now, interestingly, 2 of them were alu-alu recombinations, and they happened only between alu elements that were in the most recently diverged family. So they were very highly identical in the region, and these really look more like a non-allelic homologous recombination. The other one did have addition of a base, no homology on either side. This is probably non-homologous end joining. And this one here, we interpreted as non-homologous end joining because there was no homology on either side, but there was some microhomology at the individual join point. So then we started to see in other non-recurrent rearrangements that we started to get much more complexity. And I have to be honest that the complexity really was only revealed as we got a technology that gave us much higher resolution of the genomic rearrangement. So that is, if we sequenced the breakpoint, we didn't often see it. And if we did a back-based array, we could see duplication, and we didn't see any complexity. And actually, we use the term join point rather than break point for this specific model because of what we think is reflecting mechanism. So what we did see, so we took patients, in this case with Politzsch-Merzbacher disease, basically the central nervous system leukodystrophy, and the majority, 75% of patients, all have duplication of a region on X, which interestingly only causes the disease in males. Females who have the duplication don't have disease having to do with lionization. But when we did arrays with oligos throughout the region, rather than see, which we do still see, patients with straightforward tandem duplications, we started to see duplication, normal, duplication, normal, duplication, or duplication, triplication, duplication, or even deletion, normal, duplication, triplication, quite complexity across the region. And as long as you duplicated the PLP1 gene, proteolipid protein 1 gene, you convey the Pelizaeus-Merzbacher phenotype. Interestingly, occasionally you triplicate the PLP1 gene and you get a much more severe phenotype, and we'll talk about that in just a minute. But suffice to say, 65% of the patient rearrangements all had this kind of complexity. And some of them that didn't have the complexity, when we actually sequenced the breakpoints, and this is from the original paper, we could actually find a stretch of DNA, and then we would find another stretch of DNA which was sometimes hundreds of kb away, and then another stretch of DNA which was again hundreds of kb away, and then back at the join point. So we would see these different pieces of DNA that were juxtaposed. So it would seem to be a template-driven juxtaposition of DNA sequences that was separated by large genomic distance. And we always saw a microhomology at the join point, which we thought this microhomology might be important to mediating the joining. And so we had come up with this, what we termed fork-stalling template switching model to account for such complex rearrangements. So the proposed model is that there's a long-distance template switch between forks, and we could measure it in some cases where we had sequence of all the breakpoints. You're tethered to the original fork, and that the priming of the DNA replication is what gives you this microhomology at the join points that you observe when you're sequencing the patients from the array, where you go from transition of gain to no gain. Interestingly, when we looked at that sequence, it all fell in a region where these patients' breakpoints were non-recurrent, but we saw a breakpoint grouping at one end. And where the breakpoint grouping occurred were in these very complex sequences. Each one of these things represent 20 to 30 kb of 99.9% sequence identity. And you can not only form cruciforms, you could get cruciforms upon cruciforms. And in the copy number database of Toronto, these are some of the most highly copy number variable regions here, but the duplications take place and extend out, and when they capture the PLP1 gene, you convey the Pelizaeus-Merzbacher disease phenotype. So we think that such complex architectures, just like we saw for NAHR, there was architecture that was important, we think this is an architecture that might be important. Now, when you look at the array, you see a situation like this, where you go from no gain to duplication or gain and then no gain. You don't think it's that complex. You sequence the breakpoint and you see microhomology, so you think maybe it's non-homologous end joining. And in this one, you see the same thing. You go duplication, no gain, duplication, then no gain, but you sequence the breakpoint and you see this kind of complexity, and we call this a FOSS-test times 3. We can see where apparently 3 template switches may have taken place. But in fact, actually, is this non-homologous end joining, or is this FOSS-test times 1? So we see the same things in terms of microhomology. I think we can only be sure it's non-homologous end joining when we see little bases added or bases coming out. The microhomology really may present FOSS-test times 1. We can see the complexity better when we have duplication, because the sequences are left behind that we can sequence and see the complexity. When we have deletions, often you have the deletion, so you're missing a lot of the sequence that you really need to interpret the event that took place. Now, the next gene that we sort of looked at, we started to see a very, very frequent event in humans, and that is that we could see duplication of the MECP2 gene. Now, that's people who here work on Rett syndrome, which is due to point mutations in this gene, and girls get the Rett syndrome, but we found these duplications in boys, and these duplications turned out to be different sizes, and occasionally we'd actually find a patient that was duplicated and then had the MECP2 gene triplicated, and there shows you nicely by Fish. It's a male, so you've got only one X chromosome, the triplicated MECP2 gene, and the control from the X chromosome. Now, in this paper that's just appeared in this issue of the American Journal of Medical Genetics, when we do clinical arrays, we cover the last 10 megabases, or the 41 subtelomeric regions. And this is only 1/10 of the genome, but 1/3 of all the genomic abnormalities are detected in these regions. And this shows you every chromosome, P arm for short arm, Q arm for long arm. And interestingly, it used to be thought that 1p minus, or deletions of 1p, at the telomere were the most common, but we are finding that duplication at the MECP2 locus is the most common finding in array diagnosis of patients referred either for behavioral abnormalities, autism, or mental retardation syndromes. Now, we've studied 28 families in great detail, and all the duplications are different sizes, ranging in size from 200 kb to 2.5 to 3 megabases in size, but they all have the same phenotype, and the smaller duplications are only duplicated for MECP2, and the triplication case had a much more severe phenotype than the duplication. But interestingly, again, we saw this grouping on the one side of the duplication rearrangements. Now, when we performed high-resolution arrays, we didn't see as high of a frequency of complexity, but 20% of them had things like duplication, normal duplication, duplication, triplication, duplication, normal type sequence. And this shows you some of the complexities here of those specific rearrangements. In the ones that didn't show complexity, it's always difficult to get some of the sequence. But the first thing that we were very interested in is if we actually look at this grouping of breakpoints on the right side, and we do a statistical analysis where we take the left side and do a Poisson model for the distribution of breakpoints, which is a region of some 2 megabases that most of the breakpoints take place, where on the right side, 75% of the breakpoints take place in a 200 kb region, and actually all of the triplications within the duplication have one join point at the same exact position. And now we look at the genome architecture in that region. What's really amazing is the low copy repeat actually turns out to be the opsin genes. And these are greater than 99% identical in their sequence, and they're amongst the most polymorphic variation in the human genome. And this is responsible for color blindness in males, that we see a lot of non-allelic homologous recombination happen between these repeats. And right downstream from that is another sequence that's 11 kb of greater than 99% identity, and this can form a cruciform type structure, and this is where all the triplication ends mapped right into this region and many of the duplication ends. So we're seeing a theme, granted it's only 2 situations we've looked at in detail, where we see the grouping occurs with this complexity to the architecture of the genome, but we're dealing with more inverted type repeat structures rather than direct repeat structures, and often you can form cruciform upon cruciform. And those same sequences are highly polymorphic in the normal human population, adjacent to where we see some of these events occurring more often. So then we decided to use the proximal 17p region, looking at what was going on autosomes. And part of the problem with the interpretation of both the MECP2 and the PLP duplications is they're on the X chromosome. And the moms were always carriers. So how do we prove that what we see in terms of complexity is all a de novo event versus part of what we see on the array came as a spontaneous event and part was an inherited CNV from one of the parents? In the X-linked situation, all the moms are carriers with skewed inactivation, and we don't have grandpaternals or several generations back to really look at new events. So this was going to allow us to look at new events. And we described this as the first microduplication syndrome. After finding the Smith-Magenis syndrome mechanism, it took us about 4 years to find the predicted reciprocal duplication product, and then 7 years to bring 24 patients into the hospital for 1 week and do a battery of tests on them to define the clinical syndrome. But here nicely shows you a fish picture of the duplication and pulse field 1.1 megabase junction fragment within the limits of resolution of pulse field, everybody having the same junction fragment because of the recurrent rearrangements. Now, interestingly, the duplication, again, is less severe clinically than what we see with deletions. And in fact, part of the major problem is neurobehavioral abnormalities. And when we tested these children by both ADAS and ADIR, the gold standard clinical psychological tests for autism, they all test out as being autistic. So in fact, it was one of the first examples, this and MECP2, of copy number changes in the human genome being associated with autism. And for those of you who saw Nature 2 weeks ago, it turns out that this is the same thing for schizophrenia. Schizophrenia in multiple individuals is copy number changes around the genome, and many different portions of the genome can have copy number changes and convey that clinical phenotype. But in the Nature papers, 2 huge studies, one in Europe, one in the United States, on 4,000 patients, showed 3 regions of the genome where it happens recurrently, and it's associated with low copy repeats and non-allelic homologous recombination. So we took the non-recurrent rearrangements in patients with Piachi-Lupski syndrome, and in this case, 65% of them have complexity, where we see things, again, normal copy, duplication, normal duplication, duplication, triplication, duplication, etc. We see these complexity to the duplication rearrangement. Now, when you're interpreting these data, you always have to be aware that arrays give you copy number information, but not positional information. So they don't tell you it's coming from that portion of the genome. We always have to follow it up with FISH, and sometimes find it's actually somewhere else in the genome, the extra copy. And also, it doesn't give you orientation information. So one patient that we actually had initially interpreted as complex, having duplication, normal duplication, in fact, the genome browser, the human genome it's a good copy. I want to make sure I say that for Francis. It's a good copy, but it's terrible at many regions of the genome. And wherever there's structural complexity, it's completely wrong. And wherever there's structural variation, it only gives you one reference human genome. So in fact, our patient actually had an inversion polymorphism allele, and this array, it's just when you match the database, it looked complex, but he had this, it wasn't complex at all. It was the rearrangement took place on top of the inversion allele, and it appeared to you complex, but it's not complex when you realize the limitations of the genome browser. But we also wanted to look at a locus that we had been spending many, many years on characterizing, for which there was a lot of patient samples out there that you could screen. And after we described the CMT1A duplication in 1991, it became a commercial test to test patients with neuropathy for this common duplication. And in fact, Athena Diagnostics, and I'm not advertising for Athena Diagnostics, but they do over 4,000 of such tests a year, which always was done by pulse field to look for a junction fragment. It's a pretty complicated clinical test. But about 4 years ago, switched to MLPA, a PCR-based assay, and that gives you not only that you have duplication, including the PMP22 gene, but will tell you the size and extent of the duplication, so we can look for unusual events by MLPA. Now, interesting, the frequency of detecting duplication or deletion, so this would be causing Charcot-Marie-Tooth disease, this would be causing HNPP, is about 18%, which is a very good clinical test. But the unusual forms only occur 0.8% of the time. So at this locus, the architecture is such that 99% of the time you have non-allelic homologous recombination, you have the duplication, and that causes your disease. But let's look at the events that don't happen through the classical NAHR mechanism. And the first event we got was duplication, triplication, duplication. They were triplicated for the PMP22 gene and had a much more severe clinical phenotype. Then we also saw events that were single exon dropouts. Here's PMP22, exon 4 dropped out, exon 3 dropped out, exon 4 dropped out. And this one looked particularly funny, the data, because before the deletion, it looks kind of funny, but you don't really realize it until you do the retrospectoscope and you sequence across those areas. That event turned out to be extremely complex. And you could find sequence, and then you went to sequence on the other side of the deletion rearrangement, and then you went back to sequence in the beginning side, and in a different orientation, so it must be going on a template in the opposite direction, and then you went back to the original point, tethered, and this is the complexity, or how we attempt to explain. At every join point, there's microhomology that specifically occurs. So we could actually show that this complex mechanism, Fostes-generated type mechanism, can occur on genomic levels with sizes from 19 megabases to 3.5 megabases, or at the genic level and just duplicate or triplicate a single gene, or single exon level, and you can have only single exons evolved in the specific rearrangement. Now, this is kind of interesting to us because we think this is the mechanism for exon shuffling, and this is one of the ways you generate lots of different divergent genes, and you move different domains into different genes and create proteins. And we could only find one paper trying to explain exon shuffling since Wally Gilbert talked about it in a 1978 Nature paper. But we think this way you can take out and put in. You don't see most of the events where you put in single exons. You only have to get the exon and the splice donor and acceptor site. The other portion of the genome put in, because introns are so big, doesn't really matter. And we also think this mechanism may be important to the Ono hypothesis of duplication diversion for evolution. And that's been a predominant hypothesis for evolution of new genes by first duplicating copies and diverging them. And we think this might be a mechanism to do this anywhere you want in the genome to generate the kind of variation you need for evolutionary processes to take place. Now, the complexity of the rearrangement, if it's big enough, you can see it by fish cytologically. We described this last year with Joris Veltman's group from Holland. Sometimes you can't see it by a cytogenetic fish, but by a back array, you could already see duplication, deletion, duplication. Sometimes you don't see it by the back array. This looks like a simple duplication, but when you do an oligo level array of resolution, you see duplication, normal, then duplication. And sometimes you don't see it by even tiling path arrays. It's not till you get to sequence that you can see the complexity of the event that's taken place at the join point. So when I'm giving you estimates of the frequency of this mechanism at a particular locus, I'm always giving you the lower estimate. This is only what we've been able to see. Many of the other ones we haven't been able to work out, or because you can't make predictions like you can in NAHR, you don't know where the actual event is taking place. It can become complex to get to the breakpoints. Now, when we proposed this mechanism of replication being involved in human genomic rearrangements, it was met with some skepticism, as you might expect and like to see. At least people are reading your papers. But it was very interesting to me. I was mentioning to people I spoke with today to see this paper that has just appeared in this month's issue of PLOS Genetics. I don't know this group in France, but really showing that segmental duplications in yeast, when you design an experimental model system that will enable you to screen for a single duplication event, not amplification events, and you actually study the mechanism, you can demonstrate its dependence on pol 32 polymerase. So that this really, and half of the events were RAD52 dependent, half were independent, really showing that replication can be important to the formation of segmental duplications in the yeast genome experimental system, and we really think in the human genome it's also important. So we think copy number variation at FOSTA causes genomic duplications, may be a major mechanism for duplication CNV and a driver of the Ono gene duplication divergence hypothesis, and we also think that this may cause exon shuffling. And I think that in the context of evolution, next year marks 100 years since the publication of The Origin of the Species and 150 years since Darwin was born. And I just came back from a 2-week August vacation in the Galapagos, where I visited 3 times the number of islands that Darwin visited with my family. Here shows us straddling the equator in Quito, just outside of Quito, Ecuador. But when you see birds with blue feet and birds that can't fly but can swim, and iguana that don't live on land but eat only marine life and have made these tails that work as a flipper, and you see turtles whose shell changes because the flora that they eat is above the ground versus those that eat grasses on different islands. It's really quite remarkable to see how different species can respond to environmental changes. And in fact, work done by the grants that have been published over the last 20 years, they're 30-year studies on the Galapagos Islands, you can really show evolutionary changes even in those times associated with environmental changes, like when there was a 1-year drought on a few of the islands. And we think that copy number variation is going to be much more important to these kind of rapid abilities to respond to environmental changes than point mutations will be. So how do we incorporate this into a clinical array, a rationale and a design for the array? So if we think of the history of human genome analysis, believe it or not, it wasn't until 1956 that we even knew how many chromosomes we had. And in the 60s, we started to associate traits, Down syndrome, with difference in chromosome numbers. In the 70s, we got banding techniques so we could at least tell which chromosome was which. And in 1981, the first microdeletion syndrome, Prader-Willi syndrome, was described at Baylor College of Medicine in the Clayburgh Cytogenetics Laboratory by David Ledbetter's group. And since then, many other microdeletions started to be described because we had a better resolution of the genome. In the 80s, we got fluorescence in situ hybridization and could go to specific regions of the genome, but we had to guess where to go. As a clinician, we had to say, look at the DeGeorge region, look at the Prader-Willi region. In the 90s, we got telomere fish, and we could change our thinking from locus-specific genome management to genome-wide, looking for changes not resolved by microscopes. And then arrays came in this century, and it's not the future anymore. It's really happening. And it remains to be seen how much personal genome sequence will really get at the copy number information. So to do arrays, again, many people, but for the students in the audience, many people will be familiar with this. We array out, in this case, either clones of different portions of the genome, or in our case, more now, oligonucleotides from around the genome, and then take patient DNA and label it with one chromophore, and then control DNA in a gender-matched way, and then you can see differences in signal, the Psi3 to Psi5 ratio. Up would be duplication, down would be deletion. Remember, it's a comparative analysis, so you're not really sure if your patient has a duplication or your control has a deletion. And then, of course, you can check databases to see if these are common variants that might be benign or they actually are de novo events. And for all prenatal tests, we always do trios. We require the pregnancy sample plus both of the parents to help in our interpretation of the data. Just to show you, this is not my lab. This is not a single person event. This is our weekly array conference. The array development team is 30 faculty members. 2 PhDs in mathematics. One is required just for the statistical analysis of the data. 2 PhDs in bioinformatics. We've got 8 boarded clinical cytogeneticists that participate in this. Array development is a multi-team member thing, and we have different people who, faculty members, all contribute in their own way. The original arrays, this just shows you it, we always did dye reversals, so we switched the dye on patient and control, and here shows you the genome with about 500 backs arrayed, and now you can see a duplication, but with the dye switch it shows you what seems to be a deletion. It was 4 backs. These are chromosomes from 1 down to 22, 4 backs on the Y chromosome, and duplication of a specific region of the Y chromosome. Here shows you what a sex reversal looks. Rather than use gender matched, you use gender mismatch, so you'll see copy number change on all the X and the Y. This was important to development, chromosomes 1 down to the sex chromosomes. And here shows you what we saw right in the beginning. We could see trisomy 21 very readily and distinguish it from copy number variants, which were found in the transmitting parent, but this was a de novo event, and trisomy 21 was interpretable as a chromosome abnormality. But many of the CNVs are not yet interpretable, and many of what's in the databases called benign variants, I totally don't believe it. And we've got evidence that more and more of them have clinical consequences in our patients. So version 4 was the first one offered in February '04, where we took all of the FISH tests from the clinical lab, all the backs, put them onto one array, including the telomeres for subtelomeric FISH. Our detection rate was 6.5% in cases that had normal chromosomes. We extended the amount of coverage to other microdeletion syndromes. In '07, we switched from backs to oligos. Our rate was about here, and now we started to incorporate not just FISH tests of subtelomeric regions, disorders, subtelomeric, pericentromeric, went to different backbone coverages, switched from the back to a back emulated with oligos, now used bioinformatics to incorporate LCR design, and then increase the amount of backbone coverage. Current arrays have 105,000 oligos. We're going to 180 format in 3 months. We just added the mitochondrial genome, which comes on clinically November 1. We're finding all kinds of mitochondrial deletions and duplications. A little bit more difficult to work with. There's so many more copies of that than there are on the nuclear genome. So our design currently, bioinformatic design, is a 3-2-1 design. 3x over known dosage-sensitive genes, 2x oligos around the dosage-sensitive region, and in predicted unstable regions of the genome between low copy repeats, and then 1x is for backbone clones. And of course, we can't get any interrogating oligos into repetitive elements or low copy repeats because we have to have a unique signal to be able to interpret what it means clinically. And this is what it looks like on the design of one gene. And this is the Cornelia de Lange gene, the NIPBL gene, which is a constituent of the cohesin complex important to cystic chromatid cohesin. And I wanted to use that as a lead-in to how now we can do reverse genomics, because now at this point, as I didn't mention when I showed this here, but now we've done 17,000 clinical cases, and we're doing 150 a week. We are starting to cover most of the human genome, so now we can start to collect a half a dozen patients with deletion or duplication of a certain segment and do reverse genomics to characterize what are the clinical consequences of that structural change of the genome. And we've done that with Cornelia de Lange in a paper that just got accepted in a reverse genomics approach. So in this case, Cornelia de Lange was due to point mutations in an NIPBL gene, and it has heart defect, limb defect, and this is a constituent of the sister chromatid cohesin complex. And whether you have the actual complex constituent or you have something that helps load you onto the complex, mutations in 3 of the different subunits give you the same loss of function mutations, give you Cornelia de Lange syndrome. So now we start to look at patients with duplication of these genes. These are not loss of function alleles, not likely loss of function alleles. And we already had 5 patients with duplication of the NIPBL gene. One of them was duplicated only for the NIPBL gene. And we had 1 patient with duplication of the X-linked gene of the cohesin complex subunit. And now we can compare and characterize the facial features and other clinical features of the patients. And interestingly, you start to see this oval-type face and very subtle dysmorphic features, and this turns out to be duplication of the X-linked. These are all the duplications of the autosomal NIPBL gene. So we see that duplication gives you a clinically distinct phenotype than point mutation or deletion of that specific region. So the increased dosage causes defects in the same systems, but they involve similar systems, but they're distinct disorders. Patients with duplication of different cohesin subunit genes share similar facial features, and we believe duplication CNV of genes could be a common mechanism for many human dominant traits. Likewise, in a paper that was just accepted last Friday, we went to the LIS1 gene of region of distal chromosome 17. Deletion of LIS1 causes smooth brain. You have no gyri. It's called lissencephaly. Deletion of LIS1. If you delete LIS1 plus 1433 epsilon, another important gene for neurodevelopment, brain development, you get the Miller-Dieker syndrome, which has the smooth brain, but also has facial features, including bitemporal hallowing. So we started now to, and it's shown in mouse models that if you knock out LIS1, you get lissencephaly, but you have to knock out both LIS1 and 1433 epsilon before you'll see the craniofacial abnormalities. So we took patients with different duplications of this region, and this one patient with a complex rearrangement had duplication, triplication, duplication, triplication of the LIS1 gene. It was even more complex, but this turns out to be a CNV that comes from one of the parents. The other complexity is all de novo. Here shows you duplication, triplication, duplication. This is a straightforward duplication. Here shows you that in this complex rearrangement, the duplication, triplication, duplication is a completely de novo event. Neither parent has it, but this deletion is actually a CNV inherited from the parent. So we have to distinguish what's inherited or transmitted versus what is, oh my gosh, I'm hibernating. Can you believe that? You know what? Can you get I got the plug in there. I think what's happening is my battery just went on me. We only have a few more slides. if you can help me, here's this, and then this is the other. This is the part that has to be plugged in. If you give me the one end, over here's a plug over there. Let me see. Usually when you plug that in, the green light will go on. Actually, yeah, if we can. All that preparation, and we didn't even think to plug this thing in. It's resuming Windows. Having just gone through a hurricane and losing electricity for 2 weeks in Houston, and incredible, we got it all back, okay? So this is important to distinguish what's the de novo event versus what's the inherited event. And here shows you very nicely that we can confirm the complexity of the triplication by fish on a patient, and this is an autosomal disease, so we can see both the normal chromosome, the duplicated triplicated chromosome and the normal chromosome. Interestingly, when we do MRIs on the patient, we don't see smooth brain. We have normal gyri of the brain, but we see brain development, and the amount of cortex is different, and we can see more severe corpus callosum problems in triplication versus duplication of the LIS1 locus. We then can conditionally overexpress the LIS1 gene working with Orly Reiner's group in Weizmann Institute, and now the MRI doesn't pick up the subtle brain developmental defects, but here's with the expressed transgene, you can measure that this brain is smaller than a normal brain that doesn't have the conditionally expressed LIS1 gene, and the size of the brain and brain development is different in these animals, and actually you could show both delayed radial and tangential migration, nerve migration in LIS1 overexpressing mice, and do it by time-lapse photography during development. They're just absolutely beautiful experiments, and when the paper comes out, you'll be able to click on the movies and actually see it. So now we can take the same region that's been defined by deletion analysis and look at patients with the different duplications and assign that, and we see that you have to be duplicated for LIS1 to have the brain size abnormalities and the migration defects in the ventricular zone, whereas you duplicated for 1433, you get the dysmorphology and overgrowth syndrome as part of that syndrome. So we do think that many genomic disorders actually may be due to copy number of more than one gene insists on specific segments of the genome, but suffice to say that will take us all a lot of time to figure out. De novo rearrangements and birth defects. So we started to note that the frequency of these events, and this is from a paper published last year in Nature Genetics, where nucleotide changes in many, many studies are between 1.8 and 2.5 times 10 to the minus 8, look at dozens of genes by several different ways. Copy number variation is between 10 to the minus 4th and 10 to the minus 6th, which is 100 to 10,000 times more frequent as a de novo event. And I calculated these from prevalence data, or prevalence and molecular data, or single sperm PCR data, or recent data on array CGH trios, that is, looking at a patient with the event versus both parents. But interestingly, our calculation for CMT was between 1.7 and 2.6 times 10 to the minus 5th. Matt Hurl's group published single sperm PCR data, and for the CMT one, it was unbelievable how close the experimental data was jiving with the data calculated from prevalence and other molecular data. So how does this relate to human birth defects? How much is genomic imbalance important to birth defects in humans? And so what we did is, I should say Jinyan Liu did, is look at 648 neonates with birth defects that were referred to our clinical laboratory. And interestingly, we don't have a lot of clinical data to go on. Many are coming from the outside, So all we can go on is what the referring diagnosis was to the clinical cytogenetics laboratory. And one referring diagnosis is suspected chromosomal syndrome by the clinical geneticist. And actually, if you look at 3 published studies, and that's the referring diagnosis, 21% of the time you actually find a chromosomally visible abnormality. So one study it was 21, one it was 22, and one it was like 20.8. 3 different published studies. Now we do the same kind of study with suspected chromosomal syndrome, and in fact 68% of the time we can find by an array the copy number difference. So just going up in resolution to a genome-wide array, that's not completely, our level of resolution is probably at this point still at about 50 kb. We can threefold the detection of a specific, patient-specific de novo abnormality, not in either parent, associated with the onset of a sporadic birth defect. And this is coming out in the December issue of Pediatrics. So we do think that these mechanisms are going to be very important to sporadic birth defects in the human population. So now we have to really start thinking about human variation in terms of not just base pair changes, but genomic rearrangements. And there are endogenous and exogenous molecular mechanisms that drive both of these processes. You can get single nucleotide polymorphisms for the base changes or copy number variants for the genomic changes. And these can affect genes in different ways. And when you have the copy number variation, sometimes it may just be benign, polymorphic variation in the population. Sometimes it actually may give you disease susceptibility or resistance. So HIV infection, if you have an extra copy of the CCL3L1 gene, you are resistant to the AIDS virus. You do not get infected with the AIDS virus. Other complex traits like lupus and Crohn's are related to copy number of individual genes. Common traits, colorblindness, infertility, hypertension, autism, schizophrenia, olfactory variation, copy number variation, sporadic and inherited disease, particularly MR, mental retardation-related diseases, Smith-Magenis, Williams, Prader-Willi, Angelman, DeGeorge, Soto syndrome, Charcot-Marie-Tooth disease. Hemophilia, we've known about it for centuries in the royal population throughout Europe, but in fact it wasn't until just a few years ago that 48% of all hemophilia is one specific inversion that takes place in the factor VIII gene. And it's due to having a low copy repeat that's in one of the introns, and another copy is 500 kb upstream. So these are really responsible for significant disease. And we also have to generalize our thinking, no longer having these categories of chromosomal syndromes, Mendelian conditions, complex traits, because in fact, Alzheimer's disease has been known for decades to be associated with trisomy 21. If you have Down syndrome, you get Alzheimer's disease in your 30s and 40s, okay? But, and then last year, the complex trait of Alzheimer's disease was associated with point mutations in the regulatory regions of the APP gene, the amyloid protein precursor, changing the expression levels. And then only last year in France was it shown that duplication of the APP gene occurs in a significant number of patients with Alzheimer's disease. Likewise, for Parkinson's disease, the alpha-synuclein point mutations were associated with a dominant form first mapped in the Italians. Last year, Christine von Broeckhoven's group in Antwerpen showed that nucleotide changes in the regulatory region affected the expression of alpha-synuclein in common garden variety Parkinson's disease, and then triplication and duplication of alpha-synuclein in the last few years have been shown to be also a prominent cause of Parkinson's disease. Familial Parkinson's disease, it segregates, and in sporadic, it happens as a de novo event. So in conclusions, both mechanisms, both recombination mechanisms, non-allelic homologous recombination, non-homologous end joining, and replication-based mechanisms like FOSDES can generate CNVs. Elucidation of NAHR enabled the genome-wide predictions of instability regions and reciprocal duplication deletion events. And as we incorporate that into the clinical arrays, we start to describe many more genomic disorders around the genome. DNA replication mechanisms at some loci can account for over half of the duplication CNVs. We think any mechanism can happen at any locus, but the architecture at some locus makes you prone to one mechanism over the other. We think that reverse genomics will start to enable genotype-phenotype correlations of most of the human genome. It may take us decades to do this as we start to define more and more subtle traits in human biology. And that de novo rearrangements account for a significant fraction of sporadic traits, including sporadic birth defects in humans. And I threw this one back in there for John. I think that recombination mechanisms, we feel after 10 years of study, or more like 18 years of study, pretty confident, and this allows us prediction ability around the human genome. We're defining the rules for this FOSS-DES mechanism, but at 3 different regions now, we really see these, where the breakpoint grouping is taking place, these complex, symmetric structures that could form cruciforms, cruciforms upon cruciforms. We think it's important to establish the rules that will enable us to do genome-wide analysis and then put the interrogating sequences onto the new clinical arrays to get at this mechanism a lot better in different patient samples. And so with that, I've tried to thank most of the people involved along the way, but I'm fortunate to have terrific postdocs and students from around the world participating, and also very fortunate to have been myself trained at places like Cold Spring Harbor, NYU, sabbatical at Sanger, and in the largest medical center in the world for my clinical training. And I'd like to thank you for listening, and I'm happy to take questions. John.
In the events you're interpreting with your FOSS-DES model, these you know to be small junction repeats, right?
Well, we know that they're in the junction, but part of the FOSS-DES model also explains these large genomic duplication where there's complexity, duplication, normal, duplication. So they're not going to be in small intervals. Those are over megabase sizes.
But you infer that those two things, one is that those all have the repeats in the same orientation?
Okay, we have seen most of the time, well, we don't have a lot of data, okay, but we have seen some occasions where they're in inverted orientation that you may have copied on the opposite strand. So you have duplication, normal, inverted duplication kind of thing. We've seen that too.
And it's your belief that those with multiple junctions arose as part of a single event?
Yeah, well, that's why it's so important. Actually, even though the original paper was done on the X-linked disease, and we could show segregation of the complexity from a carrier parent, I really felt it was very important to really look at de novo events and really show the entire complexity came from the one event. That is, it was not in either parent, and you could find it in the child presenting with the disease.
As we were discussing, that means the event was completed over the 100 cell generations that went on between zygote and sperm, say.
Either that, or the event could have been completed in the opposite, where you went from sperm germ cell to making your sperm. You undergo about 10 to the 6th divisions there. It's
30 divisions.
Huh?
30 divisions. You mean
No, there's multiple.
Increase in number.
There's multiple germ cells that undergo multiple events. So we've always known that for point mutations, okay, which are causing sporadic diseases like achondroplasia, 85% of the patients, short-limb dwarfism, achondroplasia, 85% of them, it's sporadic. It's a de novo event. It's not inherited, okay? And there's 3 really well-characterized diseases, tuberous sclerosis, neurofibromatosis, and that for which new mutations are very, very well characterized. It's always associated with advanced paternal age. And the where advanced maternal age is meiotic-type driven things, aneuploidy. Advanced paternal age is associated with de novo point mutations. And
spermatocyte gives rise to 1 million sperm. You don't mean a million divisions.
No, yeah, exactly. I'm sorry. Yeah,
Okay.
exactly.
So it's 20 divisions.
Yeah, there are many replication events that can take place prior to making sperm, and advanced paternal age is thought to be reflecting that accumulation of point mutations during spermatogenesis. We don't have any data one way or the other on genomic disorders, whether they're related to advanced paternal age or advanced maternal age. There's just not enough known yet at this point.
How about,
Yes.
frequency between oogenesis and spermatogenesis
Of these
of
events?
these events?
I have to tell you, it really is a matter of what you can study. Female gametogenesis is virtually impossible to study, since female is born with all of her eggs already. This happens during early embryological development, where the male sperm are much more readily obtained to be able to study many events. And PCR allows us to study a lot more of these events at this point by using single sperm, single molecules.
In terms of the clinically relevant syndromes, where you can trace, well,
The only thing associated with advanced maternal age is aneuploidy, okay, and more recently uniparental disomy. And that's because the major mechanism for uniparental disomy, where you get two chromosomes from one parent, is trisomy to disomy rescue. So you'll have a trisomy, which would have, say, two of dad's chromosomes and one of mom's, but rather than kick out the extra dad, you kick out the mom to go from trisomy to disomy rescue. And you have UPD of the two dad's inherited chromosomes. So those are the only two phenomena really associated with advanced maternal age at this point.
These complex rearrangements, they seem very common. That is, they occur in 1 in 10,000 humans. Could you approximate?
Okay, so there's not a real lot of data on this. Okay, but let's look at the Smith-Magenis locus. Okay, there we know the frequency, the prevalence of Smith-Magenis syndrome is about 1 in 20,000. So that would say that the event is about 1 in 10 to the minus fourth. Okay, of the SMS, I showed you that 70% of them happen by the classic non-allelic homologous recombination in 100 family study. So only about 30% have this non-recurrent rearrangements, and 60% of those 30% had the complexity that we could detect. Okay, at other loci like Police-Merzbacher disease, pale duplication, it's a higher frequency of these non-recurrent. They're all non-recurrent events, and the frequency of that disorder is not very well calculated because it's a male-specific leukodystrophy. But you can get leukodystrophy by many other mechanisms. So unless a person does a molecular test, we don't have really good prevalence data on that. So I certainly feel that they're very frequent. I feel that they're very common. The data, I think, over 25,000 genes and over all of the loci, the data are pretty minimal under which I'm currently formulating that hypothesis. Okay, so we'll have to see over time and over many different regions of the genome what will happen. Certainly, our ability to detect a genetic cause for birth defects using higher resolution genome analysis by arrays really tells us that this will be a common mechanism for new mutation associated with sporadic birth defects for sure.
Okay, so they're not extremely common, but theoretically.
It all depends on what you think extremely common is. Okay?
Right.
As a geneticist, okay, something that you would call very, very rare, I'll see 4 or 5 times a week.
Yeah, but if you theoretically, all these mutations have to occur in the germline cell in order to cause the disease. So if they're common, though, people without the disease should have these events occurring in their somatic cell line.
Well, that's if it's mitotically driven.
That's true, yeah.
In the replication mechanism is seemingly mitotically occurring, but the NAHR is meiotic. Okay, so if you do a single sperm PCR assay across the CMT, H&PP loci, and you use sperm, you'll get the recombinant event because you can only PCR across it if you have the deletion or duplication the way you design your assay. And you use blood DNA from the same individual you got the sperm from, and you never find the event by PCR.
These events would never occur.
That's one interpretation of the data. Another interpretation of the data would be the assay requires that you span a recombination hotspot. That's the only way you can see it and do the PCR. Okay, so what if your recombination hotspots are different in mitotic-driven events versus meiotic events? You wouldn't see them by this assay. So that's one other interpretation of the data. But the general thought is for NAHR, it's purely meiotic. Okay, but we have a paper in the next issue of Genome Research showing mitotic events in NAHR-driven mitotic events. And the Cancer Genome Sequencing Project is starting to find many of these events by doing lots and lots of sequence on different cancers. Yes, Wolf?
This might be the same question in a different dress. I mean, some of the events you see are so frequent that they should be amenable to analysis in tissue cultures. And in particular, those events where you have them close to an architecture where one would think that it causes DNA replication problems. Have you looked there in tissue culture,
okay.
whether you could pick
We
them
have
up?
not looked in tissue culture in these, and part of that was predicated on the fact that we really only had evidence for these being meiotic, not mitotic. So we didn't really push looking at the tissue culture system. But the other problem is you can get artifacts in the tissue culture system. So for instance, okay, when they did the Redden et al. Big Nature paper on copy number variation that did 270 HapMap samples using back tiling path arrays as well as Affymetric SNP arrays, they started to find a number of the HapMap samples that had trisomy 9, that had trisomy of different chromosomes, never detected on the SNP chips before, but and it turned out that it was all because of tissue culture artifact that you saw those copy number variant type things happen. When you go back to blood DNA of the patient, you don't see it. It's because you made a lymphoblastoid cell line and a tissue culture artifact of actually aneuploidies and other chromosomal rearrangements occurring as artifacts in the tissue culture system. So, I mean, a lot of this sounds like excuses and hand waving, but the short answer is we haven't done the experiment. I mean, the experiment, I think that we need to get more and more, and certainly the reviewers of my grants, when they are turned down, think we need to get more and more into experimental testable systems. We've tended to focus on human as a model system and seeing what the actual events are in humans. But as we try to get down more and more to mechanism, we really need to have these more assayable systems, you know, like you guys are doing in bacteria and yeast. We have to get more of those in mammalian cells that we can look at. We have started to look at some things in the mouse, okay? And actually, of interest, the person I think is doing some of the best work on this has been Alan Bradley, who developed a lot of this chromosome engineering and things. And he's been going back into all of his ES cell experiments that didn't work over his 20-year history. You know, now he's been at Sanger for about 5 or 6 years, but they've taken out of the freezer everything that doesn't go germline and now do an array on these things, and you find all of these spontaneous copy number variants that probably are somewhat related to why your thing never went germline, but what you usually just did was take a different set of ES cells and go until you got it to go germline. You could only publish positive results, not negative results. So I think that we'll get more at some experimental systems to look at these mechanisms in greater detail. We're hoping to, now that we're up to 17,000, we started to find some patients that have had multiple de novo events, that the arrays picked up 4, 5, 6 de novo events, the clinical array. So we're starting to try to use these patients as a way, just like microsatellite instability was used as a way to get into HNPCC and study mismatch repair in humans to see if this will reflect some abnormality in the mechanism that's giving you a much higher frequency of de novo events in particular individuals. And so we are starting to look at this kind of thing. Yes?
Your Galapagos Island slides were very interesting. Is there any evidence that the number of copy repeats or deletions increases in response to changing environmental conditions? Would you have a de novo increase?
Well, you know, after I visited the Galapagos and really had a terrific time and learned a lot being there on the National Geographic tour, I pulled all these papers from the grants who've been for 30 years, every summer go there and collect all of Darwin's finches again and under all different environmental conditions. I would love to look at mutational mechanisms in the evolved species, but I got to first read these papers more and try to contact them and see what kind of samples that they have. And I don't know what kind of genomic tools are available in birds at this point, quite honestly. I've got to look into more of that. But the short answer is nobody's looked at this at a point to see if is it that there's frequencies that change with the events? Is it that many of the evolutionary events happen by copy number change, and since that's a more frequent mechanism, you can have the changes. I mean, if you look at Jim Sakella's work, which is really kind of exciting, where he looks at copy number of human and 12 related primate species, non-human primate species, 1/3 of all genes have undergone copy number changes, okay? And you can relate key genes in human speciation, those involved in cognition and those in developing long-distance running, which required sweating, which we have that other primates don't have. You can see those genes undergo a tremendous amount of copy number. And I don't know who I was talking to today. Another exciting story is the amylase gene. A nice paper in Nature Genetics a few months ago where the copy number of the amylase gene will change from 3 to 12 copies. You can actually study different indigenous populations of the world and show the copy number is related to what their diet is and how much starch is in their diet. And actually you can then measure not just copy number, but amylase activity in the saliva correlates completely with copy number. And that copy number, high copy number happens in high starch diet populations. Low copy number happens where you don't have. So I do think that they're starting to get more and more, maybe it's a smoking gun, maybe it's a singleton cases of involvement of evolutionary changes that are related to copy number. And actually, at a recent meeting I was at in July, in dogs, you can show that coat color that is changed in different dog species happens by an NAHR mechanism of copy number change, which was really beautiful out of the Stanford group, Greg Barsh's work. It's not published yet, but it was just beautiful data on that. So I think that we have to see over time. My gut feeling, and I don't have data for this, is that mutational rates probably won't change. They'll probably be a property of the either polymerases or recombination enzymes. Maybe, and that rate may be very different for copy number changes versus nucleotide changes. And maybe under rapid evolutionary changes, because of extremes in environment, the copy number plays in more because there was a lot more copy number variation that could have significance to be able to have such an environment, have screened out with such an environment something that can withstand that environmental change. But I don't really know at this point. Yes?
Sorry, I have to cut the discussion. We have GGG class before dinner, so I'll have to thank Jim. And, sorry, could we really go?
It's okay.
No.
Okay, it's good. It's important for the students to have some time.